Hello. Yeah, good afternoon, everyone. Welcome to Graduate Students Colloquium, jointly organized by IAS, EEE GSC, and IEEE Young Professionals. So today we have Nanyang Assistant Professor, Professor Yu-Cheng Chen, delivering an exciting talk on biolasers, exciting multicolored spectrum in PhD career. So Dr. Chen is the first student from University of Michigan to actually accomplish PhD in just like 2 years. Since 2012, he has published more than 40 journals and proceedings, while a startup company is being formed to market the optofluidic biolaser technology, which is a byproduct of his pioneering PhD work. His research group is focused on development of micro-nano biointegrated lasers, which integrate the beauty of lasers, optofluidics, biophotonics, nanophotonics, electronics, machine learning, and biomedicine. Wow. And he has achieved several breakthroughs and has invented the first laser emission microscopy that can be widely applied to biodevices, cancer immunodiagnostics, as well as neuroanalysis. So without further ado, let me call upon stage Professor Yu-Cheng Chen to deliver this exciting talk. Uh, please put your hands together. Thank you.
Thank you. Is it okay? Yeah. Okay. Good afternoon. My name is Yu-Cheng Chen. Well, thank you for the invitation to this talk. I am personally from electrical and electronic engineering, EEE, and also affiliated with chemical and biomedical engineering, and also Center of Biodevices and Signal Analysis. Today, I would like to give a talk on biolasers, which is a very cool phrase, and also exciting multicolored spectrum and PhD career. So as I showed you in my personal background, I am both affiliated with EEE and SCBE. So actually nowadays we have already entered a new generation of research in interdisciplinary field. So interdisciplinary field is the combination of different background and different research expertise. So biomedical engineering is one of the very, successful emerge of different background. So let me briefly introduce what biomedical engineering actually includes. So by including biology and medicine, we can actually integrate this concept with physics, optics, and photonics. So with the integration of these two, we can use spectroscopy or imaging tools to analyze like cells, molecules, or some disease. The integration with materials or chemical engineering, scientists try to develop different nanostructures, nanoparticles, which can be delivered into cells, bodies, to cure some cancer. If you integrate it with mechanical engineering, then the most famous one is microfluid. So people have developed all types of lab-on-chip to do some bioanalysis or even some wearable devices. And more recently, a lot of research scientists have been working on robotics. So they're trying to develop very tiny robotics which can be integrated inside of the vessels in our body, which can be cured for certain disease. Since we are EEE mostly, so if you integrate this with computer science, actually it is another very exciting field in the future. So artificial intelligence and also computer science, it actually provides a novel tool to analyze cells, tissues, patients. So we need a lot of these computational tools to analyze the subtle difference between different patients and different structures. Scientists have also used different electronics or circuits to develop some biochips. So these biochips actually are very already in the market, or actually sometimes it's in the hospital, which patient can use it to detect some brain or neurological disorders. And finally, the integration of biology and medicine with artificial intelligence is one of the most, I would say, fascinating direction, because artificial intelligence actually requires a lot of data. So data science, no matter it's the spectroscopy or imaging, from like all types of tools from the hospital. These huge amount of data can be actually analyzed and be more proficient by using artificial intelligence. So the integration of AI with biology and medicine actually provides a new tool for the future as well. So back to here, we are ourselves is in person like a machine. So machine has an input and an output. So being as a student, a PhD student or a postdoc, basically you grab a coffee and you produce papers. So that's what the basic job which we do right now. However, if you have multiple things or multiple fields which you need to integrate, for example, if you want to learn biomedicine, but you also integrate with electrical and engineering, or you try to integrate this with physics and chemistry, or if you try to integrate these with computer science or mechanical engineering, things becomes more tough because you need to learn different field, different background, you need to collaborate with different people, and eventually most students feel very stressed or either depressed. So especially if you study in a PhD working in interdisciplinary science or interdisciplinary engineering, it really requires more time to understand different field and different people. However, in my personal experience, I would say PhD, to me, I actually had a very fascinating time when I was a student. So P stands for play and have fun. So I actually did had a lot of fun during my PhD. So I would say I didn't lost anything during my PhD study. So I try to play as much as possible. And the second one is happiness. So happiness comes from a healthy body, a healthy mind, and also a healthy relationship between your family, your girlfriend, or your siblings. And D is development and research. So if you have great amounts of research developed, of course, between you and your professor, you guys develop nice research, of course you will feel, very accomplished, and then you will feel happy. So I believe in my personal philosophy, the PhD needs to have these three components in order to be a very successful PhD in the field of interdisciplinary science. So this is a brief background of myself. I graduated from National Central University, and then I went to University of Arizona Optical Center for doing exchange. And then I studied a master in photonics and electronics. And then I worked at a hospital and in a molecular imaging center for 3 years. And then I moved on to Michigan and then studied my PhD and then worked as a postdoc in the Center of Precision Medicine. So this is my whole pathway. So I would briefly go over what I have done before. So when I was an undergrad, I actually didn't have much interest in biology. So actually my research project was very purely what EEE students should do. So at that time, my project was on solar cells. So we tried to use quantum dots and some semiconductor materials to create some solar cell devices. And so that was what I did at that time. And then I had a very precious moment and did a 1-year exchange at the University of Arizona. And there you can explore the world and see the most fascinating optical center. And then when I was a master, I worked on nanophotonics. So nanophotonics means that when an optical structure is squeezed down to a very, very tiny structure down to nanoscale, many exciting properties will show up. So we try to use this nanoscale photonic structure to develop different devices. And then we use that type of devices for biosensing. So my interest towards biomedical engineering actually was developed during my master degree. So we actually use nanophotonic devices to create some tiny sensors. So when bioanalyte is attached or binded to these nanophotonic structures, you will see a shift in the wavelength. So this is a very basic concept of how biosensor works. And eventually we try to move this to some device, portable devices or some chip, in the future, which can be used. And I also had a chance to go to Paris for a very exciting conference. And then, as I mentioned, I moved on to NTU Hospital in Taipei and worked at the Medical Imaging Center. So at that time, actually, everything was out of my imagination because my goal was to do biomedical engineering. However, my PI gave me a topic which is no medical, only bio. So the topic was on dinosaurs. So during that 3 years, actually, I was forced to work on fossils and dinosaurs. So it's actually no medical, but purely biology. So we developed some nonlinear ultrafast microscopy tools to analyze all sorts of dinosaur fossil tooth. So these tooth, we got it from some museums. These are all from different time of dinosaur. Actually, I hate dinosaurs since I was young, so I don't like this topic at all. So that was something I hate so much during that 3 years, and I thought my life was, collapsing. So then eventually, so these are some results. So we actually analyze these dinosaurs and alligators. So these are really the dinosaur tooth which we got. And flipping over, these are the third harmonic or nonlinear microscopy images which we got from these dinosaurs. So we used the photonic tool to image these dinosaurs. And we study the tooth, the structures inside of the enamel and dentin. And we do a lot of analysis. And we also try to build a 3D imaging method. So we did some image reconstruction, although it sounds very scientific, right? Actually, it's not. So we use some, 3D reconstruction tools to build these 3D microscopy. And then going back to dinosaur, we use these tools to analyze, dinosaur that eats grass, that eats meat, that's combined. So these are a chart of the dinosaur eating behavior, which I don't care at all. But my advisor cares a lot. And so this is, we analyze different dinosaurs from different, age and alligator. So alligator is one of the most important one because alligator is the only animal which crosses now and before. So alligator is the most closest animal between human and dinosaur. Whatever. Finally, I still got the chance to do some medical part. So before I left the lab and went to US, I went back to study some human tissues. And then I studied some cancer on skin. So we focused on skin cancer because skin is on the outside of our body, so it's much easier to penetrate by using these microscopy. So we use the same tool to study some skin cancer tissues. And then we use some image analysis tools to analyze these structures. And we expect that we could develop some tools for some cancer diagnosis. So based on the research, I actually had a chance to go to the most famous laser conference, which is called CLIO in USA, to present dinosaur. Okay, so when I had the talk at CLIO, and I remember I finished the talk, and then when I stepped down the stage, I heard somebody say, "How come dinosaur is in this conference?" And I was thinking, that's not what I expect, but it just happened. So then, but the good thing about this conference is I actually met my future PhD advisor at the CLIO, but he was not there listening to dinosaur. So he's more interested to my CV, but not dinosaur. But something which I have to mention good here is actually when I applied for PhD program, maybe or even a postdoc program, those foreigners or those professors are very interested in dinosaur. So when I look at my CV, they don't care about all those other things I did. They just ask, tell me about dinosaur. And then, okay, dinosaur is very fascinating. I tried my hard best to do a dinosaur, and then they're very interested, and I think that's how I got my PhD. So, still, thanks to dinosaur, I got my admission to the US to study my PhD. But my PhD is, actually I did have very good admission if I go to Harvard on dinosaur. Because one professor on developmental biology was very interested in my dinosaur research. And he said, you can come to my lab to work on dinosaur. And then I said, no, I don't want to work on dinosaur anymore. So then I went to University of Michigan. I got the chance to fly to there for interview. It was very cold. Usually we have 6 months, minus 35 degrees Celsius. However, it was a very good experience. So summarizing all the things I have done before PhD, I had a background in optics. I did do a lot of biology and some medicine. I had experiences in like devices and also imaging. And regarding skills, I actually did do spectroscopy, device fabrication, and also worked on optical system and tools. So one thing which is very important before you apply PhD and before you start to study PhD is you have to be always well prepared, multiple background and skills. So it does not mean that your PhD has to be the same topic as what you have done before in your master or undergrad. However, some skills, for example, during the dinosaur research, I actually did develop the laser. I did build up the system. So those skills are very useful during my PhD. However, the result of dinosaur, of course, is not useful for my PhD, but the process through the whole thing which you have learned is very important. So it's still very good to have multiple background and skills, and ready for a new topic and open-minded is very important. So then I had a PhD in University of Michigan working on biolasers. So biolasers, I'll go into biolasers very quickly. So I actually work on biolasers. We did a lot of sensing and imaging, and we also work on a lot of devices and tools during that time. So as I mentioned, I did have a very pleasant time at the University of Michigan when I was a PhD student. I enjoyed with the time with my living, the snow, the friends, and also the pumpkin, the boating, the kayaking, and finally I graduated. So looking back, the time scale, if you look in terms of biology, I actually did a lot of research from dinosaur, leaf, DNA, neuron, cancer. If you look in terms of engineering, I actually did have a background in device, nano, photonics, imaging, tools like a microfluid, resonator, and biochips. However, these seem to be very and highly, not related. So how can you imagine that one to be related with dinosaur? Of course it's not. However, these things are all connected by my own background, which is photonics and electronics. So even though my major is still in photonics or optoelectronics, I still had a chance to really involve in all these different fields. Therefore, just a very quick note, I personally think research process includes reading, experiment, publish, and networking. So you need to read papers as much as possible. You don't really need to study too detailed, but try to look as much paper and capture the most ideas as possible from other groups. For your experiment, you need to be very accurate and try to avoid making careless mistakes. One thing which I always tell my students is you have to plan ahead and always have your backup plans. So honestly speaking, during my PhD, I did not spend much time in the lab. I would say I spent the less time in the lab. However, I graduated with the less time in the whole, the school. The reason is I actually did plan a lot, and I did always know what I had to do during each experiment. So if you do have a very, very accurate and very successful planning, each time when you do the experiment, you can actually save a lot of time during your PhD. So most PhD students, they actually do their experiment, and then eventually each week, 80% of data is garbage. So you only develop 20% of data to your PI. However, that's not very efficient. So planning is very important. And finally, you will publish. So publish step by step. you can expect to publish Nature and Science. However, you have to prepare the worst case. So it's especially for new PhD students, it's better to step by step and improving your publish. For a postdoc, I would say, of course, you need to be prepared and try to expect for the highest publication. And finally is networking. So networking is actually very important no matter you go to industry or if you go to academia. You need to make friends and you need to collaborate with a lot of people. Okay, so finally comes to today's main topic is biolaser. Okay, so here I would give an introduction of what is biolaser. So biolaser obviously has the phrase of laser. So laser is composed of 3 components. The first component is external energy source. So usually that is like this is a battery, or sometimes it's a laser pump. You need a mirror, and you need a gain media inside. So these are the 3 components of a laser. However, the definition of a biolaser is when you replace this gain material. So the traditional gain material is a piece of crystal or a CO2, for example. If you replace this gain material with biologically related materials, so that could be your cells, your DNA, your blood, your tissue, whatever. So all by putting these biologically related material inside of the cavity or inside the 2 mirrors, when you pump it, you will expect to generate laser emission. So keep in mind that here we are not utilizing this laser like what I'm using right now as an illumination source. We are using this laser as a new type of detection method. So laser, the A stands for amplification. So light amplification. So which means anything changes inside here will be amplified. So by taking advantage of this phenomena, we can amplify very, very tiny changes in the cavity. So no matter if it's a physical, chemical, or structural changes, it will be amplified, and then we collect the laser emission for biosensing and also for bioimaging. All right? So why do we need this laser-based detection compared to traditional fluorescence-based detection? So most people use fluorescent, fluorophore, to label the samples tend to achieve the spectrum or the imaging. So if you compare these two, fluorescence is a linear signal, while it provides a very huge spectrum over usually 100 nanometers of wavelength. While laser is a very directional emission, it has a pump and intensity which is nonlinear, and so it has a threshold. A threshold means that you can create like a gate to open or to turn off the signal. And the spectrum is usually very, very narrow. So you can expect a very high, strong spectrum with a very, very narrow line width. So based on these features, we have strong intensity, high contrastability, and high contrast, and high signal-to-noise ratio. So all these features can be the benefits over traditional fluorescence-based method. So we take advantage of these features to do sensing and imaging. So as I mentioned, the concept of biolaser is you can replace this gain material with all types of biological media integrated with cavity. So the cavity can be all types of cavity. It doesn't really have to be 2 mirror. You can use fiber, you can use ring resonator, photonic crystal, whatever else. It doesn't really matter. And then you can generate biolaser. So in the past, we have actually explored this field by using a lot of different molecules. So we use DNA, protein, enzyme, leaf, jellyfish protein, virus, and also bacteria to demonstrate lasing before. And the most fascinating part is cell laser. So we actually focus more on cell laser. So cell laser means that when you put a single cell and you sandwich inside 2 mirrors, and then you can expect that when the light pumps on this cell, it resonates, and then it generates laser emission. So this is a laser spectrum of a lot of cells, and this is a laser image of a bunch of cells. So these image and these spectrum are very unique. So these are the information which cannot be provided by fluorescence. However, there's another type of cell laser, which is you put a droplet or a bead inside of the cell, and then this cell itself can generate laser. So this cell does not require two mirror anymore. It can itself, be flowed through the microfluid, and this cell can generate laser emission. So we take advantage of this type of, cell laser, and then we can do cell barcoding because it provides a very unique spectrum. So these spectrum can use to identify the changes or the location of the different cells. So the basic goal in this field is to use this laser-based method to do sensing and also to do imaging. So our goal is to develop different biolaser, which can be integrated in the human body. And this is not fake, actually. One of the scientists did use some tiny laser and injected in cow eyes to pump laser emission. However, this is useless, but it's just an example of demonstrating a biolaser. So here, going into more technical part, I'm going to show how we can use biolaser for sensing. So one example is to use a fiber and putting blood inside. And we use a FDA-approved dye to label the certain parts inside. So when this dye is binded with certain protein or lipoprotein, it actually generates laser emission. So the mechanism, it works like this. If pure dyes is flowing through the fluid or the ring resonator, nothing can be detected. You will see just very clear, noise, no noise at all, background. If you integrate and flow some lipoproteins or albumins inside, when they bind together, you will generate laser emission. So this can be a very, very powerful tool to identify certain molecules, because when molecule binds together, they can generate laser. When nothing is binded, nothing is detected. So this gives you a very, very high contrast and high signal for biosensing. And we also use this method to demonstrate that actually this can be applied in whole blood. So we use whole blood to detect such molecules or proteins inside and generate laser emission inside of human whole blood. This concept can actually also move on into cells. So if you put, instead of micro size, if you put a nano size semiconductor nanowire inside of the cell. So this cell has 1 or 2 nanowire inside its body. And when the environment of the cell changes, or the inside the chemical changes, this laser will also change as well. So taking advantage of this laser that's very sensitive to environment, we can use this as a way to do sensing. So this is an example of a cell which contains like 1 nanowire inside. And then you can see that when you increase the surrounding solution with, for example, NaCl solution, the lasing wavelength will shift. And then it goes back, and then it goes forward again. So it's like a switchable laser detection inside of the cell. The next part, which I'm going to show today, is how it can be applied to imaging instead of just pure sensing. Okay. So for example, if I have 2 pieces of mirror and I put cells, or if I put human tissue inside, forms like a sandwich. Actually, the real structure does look like a sandwich. So you put 2 mirrors and a human tissue inside, and you label a certain DNA inside of the human tissue. And then you pump this tissue, and then you will expect it to resonate, and then you will generate laser emission. So this is a traditional fluorescence microscopy, and this is laser emission microscopy. So laser emission microscopy will give you a much higher resolution in terms of spectrum and in terms of image. So these tiny dots, we call it laser stars, can actually provide a much higher resolution to identify certain features compared to traditional fluorescence, like this, the cells, and this is laser. We also applied this concept to real cancer tissues. So we actually recruited a lot of patients, more than 50 cancer patients and also healthy patients. And we actually are able to identify the laser features inside of different patients. And we found that actually healthy cells has a lasing threshold which is much higher while cancer patients has a lasing threshold which is much lower. So which means there's a cutoff between healthy and also cancer. So in this way, if I pump around here, I am able to distinguish between cancer patients and normal patients. So by using this method, I can further apply to more patients to do some data analysis. So we did a lot of image, and we also did a lot of the spectrum analysis. And then we found that actually most patients do have much lower threshold if they are cancer patients. If you are healthy, the threshold is much higher because it overgoes a regular cell cycle. So then we can use this method to develop a new type of imaging. So as you saw, that it's a two-piece of mirror, and then if you scan it across the whole image, then you will be able to form a new type of a large area imaging method. We call it the laser emission microscopy. The reason why we call it laser emission microscopy, because what we collect is laser. We're not collecting fluorescence emission anymore. So if we can see that normal patient, usually almost nothing, while cancer patient has a lot of lasing emission. So you can see the sensitivity is about 97.5, and the ROI curve can provide an area of 0.998, which is perfectly well. So this is a basic comparison between fluorescence and laser. So if I have two fluorophore, or I have two dyes, which has very, very similar wavelength, for example, FITC and YOPRO, doesn't matter. So they are very similar in terms of fluorescence. If you label the same patient, you will see that the images look similar because they all present green. However, if you put this inside of a laser cavity and then you excite it to generate laser emission, you will find that actually laser emission is very unique. So these two dyes can be separated perfectly without no overlapping in terms of wavelength. And the images are actually very unique and very different. Of course, there are a lot of physical information back beyond this. However, this is just to show you that laser actually has a very unique method to provide a unique spectrum to distinguish between traditional fluorophores. And the image part can also provide very unique information other than traditional fluorescence microscopy. Next is the third application, which we would like to show, is how this laser can be used for tracking and recording. So as I mentioned, this laser can not only be created with two mirrors, it can also be created inside the cell. So if I have a very tiny semiconductor nanodisk or 1 microdisk, which is small enough to put inside the cells, and then this nanodisk can provide one laser spectrum. So each cell will provide one unique laser spectrum, as I mentioned. So they don't overlap with each other, and they can have a very unique laser spectrum. So if I have a bunch of cells, for example, if I only have 1 cell, 2 cells, 3, or 6. So if I have a lot of cells, or if one cell has a lot of beads, I can actually use these spectrum very uniquely to identify where the cell is going and where the cell is circulating inside the tissue. So this is example by putting these semiconductor nanodisk inside of a mice, and then you can track in the tissue which wavelength versus the location these particles are going. And of course, this can be integrated inside of tissue. So if you have a tumor spheroid, a tumor tiny tissue, which actually grows over time, each cell has one semiconductor nanodisk, for example, then you can track all these cells over several hours, and you can identify by the color, because each color is very different by wavelength. And you can see how these cells inside a piece of tumor migrate over time by using this laser method. And then you can track it at a different speed. So then you can identify by different sizes, wavelength, and you can track these cells by low mobility or high motility. Finally, we would like to show how this can be used as a device, or how it can be used by integrating with neuron. So, for example, if I have a piece of two mirrors and I put a neuron inside, this neuron is labeled with certain chlorophyll. And we all know that neuron has action potential. So when the neuron is excited or is not excited, it actually does have a certain, contracts, which can provide, like a heartbeat profile. So here, we would like to solve the traditional problem which fluorescence has met, which is the contrast is too low. So we put this neuron inside of the mirror, and then we excite it to generate so-called the laser emission image. So when the neuron is contract, it generates laser. When the neuron is released or relaxed, there's no spiking behavior. So based on this, we can put a CCD on top over time to record the behavior of neuron by recording the laser behavior. So, and then we can identify the different intensity over time. So besides doing this in one neuron, we can also apply this method to several neurons and also multiple neurons in a big neural network. So by expanding the field of view, we can actually track these neurons, where it moves and how the action potential is changing. And we can improve 1,000 times of sensitivity and contrast over traditional fluorescence. And if you put each neuron inside of a micro well, and then you scan it, of course, you can do single cell analysis, and you can identify each neuron. And this is very powerful because this can be integrated with some drug testing for patients. And this is an example to show you that during each well, for example, over a different time, the laser image actually changes, and the spectrum also changes. So laser is very special. It actually provides different information, like compared to traditional fluorescence. The laser image is very related to the changes of the cell or the structure. And the spectrum is also very sensitive to the chlorophyll you labeled. So these information create a huge amount of data which needs to be explored in the future. Actually, we don't know quite much about the behind information of these yet, because this is a very new field that we are also trying to explore more. So finally is my conclusion. So very briefly, I actually showed you several applications by using this biolaser. It could be inside the cell, it could be used for blood, for imaging, tracking, recording, and also by developing into a device. It has the potential for high sensitivity, high contrast, narrow line width, high resolution, and lasing threshold. So it can be integrated with a lot of different fields in terms of optics, biology, and biomedicine. So if we look to the very close to the near future, actually, when you put a single cell or something inside the cavity, this thing, in summary, can be replaced by cells, protein, DNA, RNA, membrane, or single molecule. And then you can record it. You can record it. So actually, in this case, what you generate is electrical information. However, if you change the bottom structure, you can try to record some mechanical information or chemical information or magnetic information as well. From the top, this provides a spectrum. As I mentioned, this laser provides a very unique spectrum. And from the bottom, you can collect the image. So you can see that by this structure itself, it amplifies a lot of tiny signals inside of this biomedia. And it actually creates very unique spectrum and really unique images, which relies on the future to do some analysis or machine learning in order to integrate and understand more about how these information tell you. So if we are able to distinguish between these spectrum and how to create these data from lasing threshold, and also these images from these laser mode profile, it actually provides a lot of information which you can use to create biological network, and you can apply some machine learning or deep learning methods to identify different compounds, genes, functions, cells, and even disease. So what we call this is it's a new type of multi-parameter analysis, which can be integrated for a new type of method for identifying cancer and neurological disorder. Finally, outlook is, as I mentioned, biomedical engineering is moving very fast. It actually is not simply just belong to medical school or like biology. Inside of each biomedical device, it actually has a lot of components. So from the very beginning is a sensing chip, which relies on biochemical scientists to design the chip and how to accurately sense different protein and biological medium. And then it relies on electrical and mechanical and also optical methods to how to amplify the signal which you detected. And then after you collect these signals, how do you process and recognize, identify so many data information, and how to use this to create a circuit which can be eventually built up into a machine. So for each machine, nowadays, it actually requires multiple background, multiple knowledge, and multiple people to collaborate and to learn between each other. So for example, if I want to build in the future a biolaser device, these different background and knowledge can all be applied to integrate into this thing together. So finally, as a conclusion remark, I would like to give fellow students or fellow postdocs actually biolaser, which means, you know, bio, people is also bio. So PhD students or postdoc, you guys are inside of an optical cavity. You guys doing experiment very hard. You guys are pumped by your supervisor. So your supervisor gives you stimulation to trigger you. And then your cavity, which is NTU, your topic, your resource, and your lab peers. And something which is more important is your own mind. So a healthy mind, a happy mind, and a very good attitude. And very good research plan is very important. And then after resonate, you can graduate or find a very good position after, you know, outcome. So laser compared to fluorescence, I would say very different in terms of people or in terms of future. Fluorescence is broadband and very low signal. So we don't need you to have so many, you know, skill sets and know everything, but just a little. You need to be strong enough, like laser, but proficient enough so you have a narrow line width. So what I expect my students or future students, after they graduate, you need to be strong and can be very proficient, like a laser emission. And not only one wavelength, but also multiple wavelengths in order to achieve a bright future. And finally, I would like to thank all of you guys for attending, and we welcome all different types of students to join our team in NTU. Yeah, thank you.
Yeah. Okay. Yeah. Thank you, Professor, for this interesting talk. And we'll take questions now. Yes.
Yeah. Thank you.
I have two questions.
Okay.
Question number one, what you call biolaser emission, I think it's stimulator Raman that you're observing.
No, it's different.
Stimulator Raman?
No, it's different.
You don't think it's stimulator Raman?
No, it's not.
Your lines and all that, they are vibrational lines, you know?
But Raman is label free. We label it. So this spectrum is determined by the fluorophore you label.
It is?
Yes, it is.
It's also, it depends on the fluorophore.
Yeah, but stimulated Raman is similar, but the effect is different. There is a paper on putting the Raman into the cavity, but I think the performance is still different because Raman is more by vibration.
Yeah, vibration.
By different like chemical bond or whatever. Yeah. But here is not vibration because the signal is, although I always do like, you know, resonate, but it's not the vibration.
But those frequencies are all vibrational frequencies.
Oh, it's based on optical resonate. I don't think it's.
But they're all vibrational frequencies. You will look at that.
Which one?
Yeah, all your spectra are vibrational frequencies.
But Raman is identified by, you cannot change the spectrum, right?
No, you are doing actually stimulated Raman where you have a pump and then there's a probe going on inside your laser cavity. And
We don't, but Raman has two probes, two lasers.
No, you can do spontaneous with one laser, just a pump.
But here, this spectrum, we can totally design it. We can change, because, for example, this wavelength, we can shift it to nanometer. We can shift it to visible as well. So for a same DNA, we can make it into green emission, red emission, or blue emission. But the Raman, it's basically fixed by the sample's original feature itself. It's more oriented by the biospecies.
So what is causing all these sharp lines? I think they are vibrational frequency.
No, because this we label it, right? And then the fluorophore, the dyes inside is resonated by the cavity. So you can imagine it's kind of like a dye laser. Yeah, so it's the dye inside the biological medium, which is we wouldn't call it vibration, but we will call it resonating back and forth to amplify the signal.
Okay, maybe it's the terminology.
Yeah, but I would say it looks similar. Yes, yes, yeah.
My second question, when you work on the dinosaur, you showed the principal component analysis, right? The two-dimensional plot.
Yeah.
Now, our birds were said to have evolved from the dinosaurs.
Yes, that's another way of saying yes.
Where do they fall on your plot? And how about humans? Where do they fall?
Let me see. Okay, you mean this one, right?
Yeah.
Some people say chicken. Chicken is the most closest to the flying dinosaur. Alligator is the one most closest to the reptile, and then most to the land walking dinosaur. Human. I forgot. but we did a lot of human, yeah. But the research is on dinosaur.
Sure, we can have a few more questions.
Thank you.
Anyone? Yes. Is it on dinosaurs? Is it on dinosaurs?
Okay, thanks for the presentation. I just want to ask how to control the number of the nanodisc in each unit cell. Because if the fluorescence material is homogeneous distributed, it's somehow difficult to achieve the laser stimulated emission.
Okay, so currently we have no ability to control the amounts of nanodisc inside of each cell.
Then how to achieve the aggregation or something? It's not homogeneous distributed, right?
You mean the aggregation of the nanodisc, right?
Yes.
Okay, so each nanodisc is not purely a nanodisc like it's like this. It actually has a silica coating outside, so it's not pure semiconductor nanodisc. Outside has a shell, and actually it's treated by some functional material. So trying our best to prevent some aggregation and clogging.
So what's the size of the post-treated nanoparticles? The size?
You mean the total size of this? Usually 1 micron.
So after the post-treatment, the size is about 1 micron.
Yes.
However, for the 1 micron, how the particles come into the cells?
First, by these type, you choose the type of cells which can eat or endocytosis.
1 micron.
Yes.
Really?
Yeah. Because even even the nanowire one, although you see the nanowire is called nanowire, it's because the diameter is nanowire, but the length is not nano. The length is actually usually 2 to 5 micron. But the cells still endocytosis this nanowire itself.
Okay.
No, actually, it's like this. It depends on what type of cell. So, like a macrophage is very like a hungry type cell, so it eats everything. So no matter how big and how small, the cell will eat it, and the cell will increase its volume as well. So they eat more and then they grow larger. However, like a neuron cells, they are mostly fixed. They don't grow like super gigantic, you know. So like neuron cells, they don't eat this nanowire. Yeah, so this is actually dependent on the cell type.
Okay, thanks.
Thank you.
Thanks. Any more questions? Sure.
Thank you, Prof, for your interesting lecture. I would like to understand how you are pump the biological tissue within the mirror. So you saw in one of your slide that you have a mirror, and within the mirror you put some biological cells or tissue or DNA, whatever, and you say that you pump that to get the laser. I would like to know what are the pump, what are the process to pump the tissue, and how tissue are generating laser.
Okay, so this, you mean the position first? You have several questions, right? One is how can I make sure identify the pump location, right? Okay, so for cell, because the laser beam is usually smaller or a little bit bigger around here, the size is very similar to the cell. And because usually when we pump it, we have microscopy, so we can actually very clearly to see where the laser beam is shining on the cell. However, if it's molecule, you're right. For molecule, if it's a lot of molecule, of course you have no ability to visualize it. So for molecule, it will be like more like solution based. We cannot, so far yet, we cannot identify single molecule yet. So when we measure the molecule part, it's a bunch of molecule which you label. It's not single molecule yet. Yeah, but that's the future. Yeah, we're trying to improve. Yeah, thank you.
Okay, with this, I would
One more
like
question.
to... one more. Oh, I'm sorry.
Okay.
Oh, sorry.
Yeah, for that cellular-based biolaser, for me, the spectrum output spectrum is much similar to the random laser mechanism. So can I say that the output is due to random lasing mechanism or a different mechanism? The laser output, the mechanism responsible for that for this laser.
You mean this one?
Yeah.
Because it's a lot of peak, right?
Yeah.
Okay, so this peak is includes a lot of cells. It's not single cell. Yeah. So it's multiple cells. But for each, if you only shine on one cell, you actually can. It's still multi-peak, like this. So if it's one cell, usually you get less peak. But this one is a lot of cells, so you get more peaks. However, the number of the peak is controllable. You can change your pump intensity. So the higher you pump, the more peaks you will get. The lower you pump, the less peak you will get. Yeah, thank you.
Okay, so with this, let us thank Professor Chen again for this very interesting talk. And the next talk would be on 13th September. And the title of the talk is Academia versus Industry. It will be chaired by Professor Avi Mendelsohn, who is from Technion Israel Institute of Technology with over 30 years of experience. So yeah, look forward to seeing you all over there. And refreshments would be served outside, so please help yourself. Thank you everyone for coming. Thank you.